thanks to, work laid down ahead of him by Dr. Montine, and then really Dirk Keen really took the baton and has generated, I think, one of the country's top brain biorepositories. So just a little bit about who we are, what we do, and then most importantly, how can we help you guys in your research studying Alzheimer's disease and related dementias. So I already mentioned we're led by Dr. Keen, and then Jessica Young and I are our co-leads. So as I said, I'm a neuropathologist. Jessica Young is a stem cell neuroscientist. And then we have a number of co-investigators and consultants, including Edleen, who is an Allen Brain Institute investigator. Amber and Desiree are other neuropathologists here in our department, and Martin Darvis is a neuroscientist in our department. And then we briefly introduced Erica and Amy, who are probably the most important people for you guys to know on here, because they really are the interface between the neuropathologists and neuroscientists and you guys, particularly Amy. She will be the one fielding tissue requests, and so they're both on here and can help me answer more of the nuts and bolts questions you guys might have throughout or at the end of the talk. So that's who we are. What we do is we support brain aging, injury, and neurodegeneration research. And we do that with some basic aims as part of the UW Alzheimer's Center. So the first and foremost is that we are a highly accessible biorepository. And we are one of the oldest in the country. So we have a really large amount of biospecimens available, and we are excited and willing to share those biospecimens with you. One of the things that's nice about our core is it is run by a neuropathologist, and so all of our cases are reviewed by board-certified neuropathologists, so you can feel pretty confident in the diagnostic variables that are associated with the tissue that you're requesting from our core. We boast a pretty rich training environment, so we're excited to be a part of the new rec core of the ADRC and be helping in that mission. We help train the next generation of neuropathologists as well as other neuroscientists. We develop innovative neuropathological approaches. So the science changes, we learn new things, we learn how to do new things, and we want to make sure that the tissue we have available is amenable to all those new approaches. So we're always trying to adapt how we collect the tissue and then how we are able to use that tissue to really push the science. Through Jessica Young and the stem cell research that we do, we foster mechanistic research, so I'll get into that a little bit. And then ultimately this is to all promote local and national research. So those are our aims, that's what we try to do, and you should not hesitate to reach out to us, because this is really what we're here for, to help you do your research to better understand Alzheimer's disease and related dementias. So you have a sense of some of the studies that our core supports, so the types of cases you might be able to get from our center. So across the country, there are some 30 Alzheimer's centers with neuropath cores, and they all have their own flavor. Yes, we're all going to have Alzheimer's cases because we have an Alzheimer's disease research in our clinical core. Here, we really boast a strong neurogenetics component that Tom Bird has been a big part of, and now Sumi Jayadev and Kimi Demoto-Riley. They bring in really interesting neurogenetics cases of all different types. So this is where we get a lot of our dementia subjects, because these people come to clinic with a specific neurological complaint. We also have population-based studies. So these people are not presenting to clinic, but they're sort of being plucked out of the Seattle population when they reach a certain age, and they're followed clinically and have the opportunity to donate their brain when they die. And so that includes the adult changes in thought study, the Seattle longitudinal study, and then we have two studies or two other flavors of studies that come out of the VA, the protective variant study, and then a number of Parkinson's disease or other Lewy body disease related studies. So that's kind of the bulk of, I think, what this group will be primarily interested in as far as aging and neurodegeneration, but we also are really ramping up our biorepository for traumatic brain injury and chronic repetitive neurotrauma cases. So we have a few studies that bring those brains into our center. And then we partner with the Allen Brain Institute, who's trying to map normal brain. And so we try to collect, quote, normal human adult brains for them. So that's the bulk of the types of cases. And then you can see here, this is just a summary of the type of pathologies that are present and how that kind of, how that tracks with whether the subjects are demented or not. So because we not only have our Alzheimer's center cases, but we also have these population-based cohorts, we have a lot of non-demented subjects in addition to our demented subjects. So that means we're really well equipped to have, you know, these case-controlled kind of studies where you want X amount of AD cases and some matched normal controls. We can do that. And then the pathology is really variable. So the blue here is the Alzheimer's type neuropathology. So there's a lot of Alzheimer's type pathology, and then vascular brain injury and Lewy body disease, and what will be added when we retrospectively go back and assess TDP-43 pathology, that'll be on there as well. So just a really nice distribution of non-demented, demented subjects with a wide range of pathologies. So we're really well equipped to answer a lot of different types of questions. So I will warn you, I do have some somewhat graphic photos. I'm going to walk through our processes just because I think it's somewhat helpful to understand how we have built this repository and the lengths that are gone to acquire this tissue. We have a fantastic team of on-call technicians that pretty much have to be ready to go at a moment's notice if a subject in one of our studies dies. The idea is we want to get that tissue as soon as possible, obviously respecting the families and their grieving processes, but ideally we have a tissue within hours, have a brain within hours of death. So the longer the brain sits, the tissue just starts to degrade. So for high quality tissue, shorter postmortem intervals are ideal. So I'm gonna have some photos of gross brain tissue here. So if we get a case within our required postmortem interval of about 12 hours, we'll do what we call a rapid brain autopsy. That means we're going to split the brain in half. One half is going to be processed fresh. We're going to select out specific brain regions that are frozen, so then we have tissue available for techniques that require fresh frozen tissue, like molecular analyses, biochemistry, immunoassays, that kind of stuff. Then the other half we fix entirely in formalin and section once it's fixed, and that tissue is available for standard neuropathological techniques that require fixed tissue, which is most commonly immunohistochemistry. So ideally we get a rapid case and we're able to get both the fixed and the frozen tissue. If we can't arrange a rapid autopsy, the entire brain will get fixed in formalin and they'll all get these nice fixed sections. And there's still a lot you can do with fixed tissue. So this is historically, and I say historically, it was up until, I guess, really 2018, 2019 that we were doing this, and we'd done it for, I think, since early 2000s. When we did a rapid brain autopsy, it meant that the technicians had to actually go into all these different brain regions and sample out a cassette-sized amount of tissue and snap freeze it in liquid nitrogen. So we would take 5 sections of midfrontal cortex and inferior parietal lobule. And we had predetermined these regions because these seemed to be regions that people were most interested in studying when studying neurodegenerative disease. You can see things like hippocampus and amygdala, and all the temporal lobe is on here. So it's a really great resource because it's easy for us to fill those tissue requests. If you say, hey, I need 10 Alzheimer's cases and I want their frozen midfrontal gyrus, we go in, we grab some cassettes. It's pretty easy to fill. The problem with this method was twofold. The snap freezing in the liquid nitrogen, while that's great for a lot of the assays that you do with fresh frozen tissue, kind of the grind and bind approach, it wasn't as great for some of the more sophisticated approaches that needed the cells to be intact and have good cytoarchitecture for single cell profiling techniques. So we needed to come up with a better way to freeze the tissue. The other problem was, as the science changes, as the questions change, people might be interested in different brain regions. And so just having this set predetermined brain regions, we realized wasn't good enough. We weren't going to be able to serve the research community well enough doing this. So we partnered with the Allen Brain Institute, and they had a very specific way that they did their freezing protocol. And after evaluating how they did it and how that could fit into our workflow, we realized this was the way we wanted to move forward. So since 2019, all of our brains are using this sort of precision neuropathology approach. And so instead of pre-selecting out brain regions and putting them in liquid nitrogen to snap freeze them, we now section the half brain at rapid and freeze whole slabs of brain in this isopentane, supercooled isopentane. And so then you have samples representative of the whole brain at your disposal in the freezer. So if someone comes and says, hey, I want orbital frontal cortex, now we can actually provide that frozen tissue for them. The other nice thing is we only freeze every other slice because we cut them very thin. And so if you have questions where you want to use a modality that's better suited for fixed tissue, but you want the adjacent tissue to do frozen work, we actually can provide the tissue to do those kind of projects as well. So just again, a brief demonstration of how our techs do this. They select the half of the brain that they're going to process rapidly. We remove the thin leptomeninges, so the fibrovascular coverings of the brain, which just sort of tear into the soft brain parenchyma. And it's really challenging to cut a fresh brain, so we try to make this as easy as possible for the technicians. So get rid of the leptomeninges, and then we place the brain and hemibrain and hemicerebellum onto scaffolding in a box. And we place it the same way every time, so every brain is cut exactly the same. And then we fill this container that the brain is scaffolded in with dental cement. So this is alginate. The kind of stuff the dentist uses to make a mold of your teeth is what we use to keep the brain nice and tight and easier to section. Because if you've ever tried to cut a fresh brain, it's like cutting super soft tofu. It just wiggles and jiggles all over, and it's really hard to get nice cut sections. But this whole block of alginate with the brain inside solidifies, and it will do it at room temperature or colder, so we don't have to worry about heating the brain, which would not be good for a fresh brain. And then we put that whole block of alginate plus brain into our slicing sled, which is really nice because it has a sliding mechanism, and you slide that block out as far as you want to cut it, so you can cut every slice the same thickness. We move ours to 4 millimeters. And then there's a guide for your blade. You come down, you slice a nice, thin, even section of brain all the way from anterior to posterior. And then this alginate just peels off. You can see we have, slices all through the brain. I think we get around 50 slices on a normal adult human brain, and then every other slice will get frozen. So to freeze it, we place it on these Teflon coated metal plates and place that plate in the isopentane that's been supercooled with dry ice. We put another plate on top that allows the brain to freeze evenly on both sides and helps keep from sinking of the white matter, which freezes at a slightly different rate usually than the cortex, so that when we take it out, it's this nice, smooth, still nicely intact, frozen piece of brain. And so this gets put in our freezers after it's vacuum sealed, and all of this gets photo documented. So then if we get a request that says, hey, I need some superior temporal gyrus, we go to our database, select the cases, we can pull up the photo documentation, say, okay, we're gonna go in and take this piece of superior temporal gyrus, and then you know exactly which slice it is because they're all numbered. And then the team can go in, pull the slice, and sample the tissue rapidly so it doesn't thaw, and we can get you your frozen tissue from exactly the region you requested. So that's been a recent advance in how we process our tissues, and we don't have that on every case going back the duration of our center, but going forward, that's what we'll have, and otherwise we have those nice tissue, those pre-selected regions. Another advance that we have made recently, again within the last couple of years, is ex vivo MRI, which is now performed on every single brain that comes through. And this has really been led by Dr. Christine McDonald, who's a neuroimager and part of the biomarker core in the Alzheimer's Center. And the reason for doing ex vivo MRI was really several fold. So the first is that you can correlate with the antemortem imaging. It's almost like a stepping stone between antemortem scans and slabs of brain tissue. It can help you link up a scan that maybe was done 5-10 years before the person died with what their brain tissue looks like now. It also helps you identify lesions that are not visible grossly. Neuroimagers are always commenting on weird stuff that they can see that if I look at the slab of brain, I don't see anything wrong grossly. But if a neuroimager can guide me where to sample, then we may find something histologically that's abnormal or different about those areas that they're seeing on their imaging. We can use the neuroimaging to help us more accurately sample specific regions. So we take, for example, the motor strip. We want to sample that in a case of ALS. Well, sampling motor strip sounds easy, but it can be really hard to identify your pre- and postcentral gyri when you've sectioned the brain coronally. And so having the imaging can help identify which coronal slab that the gyrus of interest is in. And then finally, quantitative volumetric analysis gives us way more information than a neuropathologist saying mild, moderate, severe atrophy, for example. And so they run the same quantitative algorithms that they would run on an antemortem scan and can give you volumes of the hippocampus or the amygdala or different cortical lobes. And so before we were imaging every brain, we were still processing them very similarly. So fixed brains are allowed to fix for about 2 weeks, and then we embed them in Agar. So Agar does need to be heated, so this has to be done in fixed brains only. It won't work in the fresh brains. But then you have a brain in this nice block of Agar, which again, just like in the fresh brains where we had the block of alginate, it helps keep the brain all together, and it's much easier to slice this way. And we can color code our Agar. So if we're doing a bunch of brains and getting them ready for imaging, we can tell them all apart based on their colors. And then that's Christine and her crew actually taking the ex vivo brains and putting them in the MR scanner to get us nice ex vivo MRIs. And so she provides us with her interpretation of some of the pathology that she's seeing from a neuroimaging standpoint. And like I said, we can kind of use that to match up with the coronal slabs that we have. And I wouldn't necessarily have sampled this region, but we wanted to know what we were seeing, what you could see histologically based on what Christine was seeing on the scan. And then I mentioned you can link it up with the antemortem imaging. So this is from an individual who had a lot of chronic repetitive neurotrauma while they were alive that resulted in getting a lot of antemortem scans. And so one of their scans in life, which was several years before they died, showed this pretty striking lesion in their frontal white matter. And then if you jump over here to the slab, we don't actually see any lesion grossly. And Christine didn't actually see a lesion on her ex vivo scan, but she was able to register her image to this image, which served as sort of a linker to being able to register to our coronal slabs so we could find the exact spot to sample. And then sure enough, there was a lesion in that white matter that suggested there had been some kind of traumatic event there that caused a lot of bleeding and loss of myelin, and what was left was just sort of a few macrophages and blood products. So there's a lot you can get out of the MRI images, and we work with Christine, who has all the scans, but she shares all of the volumetric data. And so if you had a project that you wanted to do some sort of imaging neuropath type of correlation, you would work with us and we would bring Christine in and help set all that up. So after they get imaged, we section our brains. And the reason we're able to do this really nice neuroimaging neuropath correlation is because we cut our brains very consistently and thin the same way every time on our brain slicer. And that's this guy here, which is really just a deli meat slicer that we have adapted to slice our big block of Agar and brain. But the result is we get these really nice thin sections. It's really hard to do consistently if you're cutting by hand. I mentioned too that our Neuropath Core has great diagnostic expertise. We follow all the latest guidelines in assessing the brains for the neuropathology. And so we sample a certain set of brain regions, the fixed tissue. So we have all of those blocks available. So if you needed frontal cortex, we always have a block of frontal cortex that we can cut unstained slides or stained slides for a study. But then we realized, we're really selecting the brain regions based on this assessment of neurodegenerative disease. But there are many other brain regions that people may be interested in. And certainly over the years, we've gotten requests for many different brain regions. And it's much better for the tissue to be in paraffin than sitting in wet formalin for a long time. So we decided we were going to use this modular sampling strategy and just upfront on every brain collect over 85 samples. This is all the paraffin fixed tissue, which we have bilaterally. And it's sort of modular in the sense that, we have all of our standard neurodegenerative diagnostic blocks. We have all the brainstem blocks. We have blocks that are specific to chronic traumatic encephalopathy. These are regions that field has felt critical to assessing a brain for CTE. Brain regions that might be relevant to severe neurotrauma, and then brain regions that have been identified based on neuroimaging connectivity networks. And so we don't routinely cut and stain and look at all these sections, but they are available. And so almost any brain region someone is interested in will already have the paraffin blocks in existence. And so we won't have to go back to the bucket, as we say, to find the wet tissue region that you're interested in. And then I think this is the last sort of resource page that I wanted to go through. And this is our stem cell resource. So Dirk really got this up and running in our core, and then Jessica Young now is kind of the person keeping up that arm of the neuropath core. So what we do at autopsy is we actually remove a strip of the leptomeninges even before we take the brain out of the skull, and we cryopreserve that strip of meninges. And the meninges have a lot of connective tissue. So there are these fibroblast-like cells that are in that tissue. And what we have found is you can actually take those cells and generate primary cell cultures that are very much like fibroblasts. And from those create pluripotent stem cells or direct convert to your cell of interest, like neurons. And so this is where we're contributing to that more mechanistic type of research, because the cool thing about these cell lines is they link back to the individual that they came from, whose brain we have, whose pathology we know, whose clinical history we have a sense of. And so it's just a fantastic resource for kind of moving to a more mechanistic approach instead of the sort of qualitative, descriptive analyses that we do more routinely in neuropathology. So this is just a summary of our resources, and this is constantly going up. This was one of the more recent slides that I had, but we have thousands of fixed brains, and that's led to almost 60,000 paraffin blocks and over 40,000 frozen cassettes. Now we have our frozen precision slices, so we have over 2,500 of those, which means we probably have close to 100 brains that have been processed in that manner. We've collected meninges on over 300 cases, and from those generated cell lines on 40, with some going to pluripotent stem cells and others going to neurons. So this is sort of a broad catalog of what we have and the kind of resources that you can request from our core. And then this is just sort of a summary of the types of applications, but really your imagination and your questions are the limit to what you can do with this tissue. And we're always happy to have discussions with people about, if they need a different type of preservation method or different types of tissue, different brain regions. We're always happy to have those conversations and find out what is needed, what is the research community looking for, because again, that's one of our main purposes, if not our main purpose, is to help support Alzheimer's disease and related dementia research. So that's what I have on our resources. I don't know if we want to take a minute if anyone has any questions specifically on that, or if I should move now into the nuts and bolts of making a tissue request, which this is pretty short and straightforward.
I suspect that we will have a lot of questions, so let's push through the nuts and bolts and then open it up for more broad questions after.
Okay, great. So I showed you a lot of stuff, and I apologize if it was a little more granular than people cared to see, but I just think it's important for you to kind of know what goes into these tissue collections and what we have available. To use it, the first thing that really gets you on our radar as someone interested is to fill out our tissue request form, which is online, and I'm going to show an image of that. So if you fill that out, that goes right to Amy Shantz, and she is going to review the request and bring it to the neuropathologist. And we meet on a weekly basis and go over these tissue requests, not so much to judge your science and whether it's worthy of sharing tissue with you, because we don't feel that we are the ones to judge that. It's more to figure out how can we best help you, what resources do we have, and how can we help you. And then if we need further clarification, then we might reach out to you. So if you're filling out this online tissue request form and you're just like, I don't really know what they're asking, I don't know what they want, I don't know what I want, just fill it out to the best of your ability. And we aren't going to trash a tissue request because we can't figure it out. It's just going to stimulate us to reach out to you guys and maybe set up a call and have a conversation about your study, and we'll offer our guidance on case selection and study design at that point. If you're not within the UW system, you may need to set up a material transfer agreement. I don't know if we have anyone non-UW here, but we share tissue with people all across the country and even throughout the world. So it's not a hard thing to set up an MTA, and Amy can speak more to that. And then at that point, once we sort of know what you guys need and where it's going, we identify the relevant cases and tissue resources, and then coordinate the material transfer. And then really all we ask in return is that the core is cited in any publications that result from use of the resource, and that there's some reciprocation of data as applicable. So sometimes if molecular data is generated, it might be helpful to have that back in our system to reduce reproducing some of the same studies, always appropriately annotated and acknowledged. And so that's the basics of how to make a tissue request. Here is where you would go to find that online tissue form. So that's really what'll kick this off. So you go to the Alzheimer's Center main webpage, and you can see here there are several links, and you want to click on the About link. And then from there, you can see there's all the cores listed here, and we're the Precision Neuropathology Core. So you click on that, and that'll bring you to the Precision Neuropathology Core page. And there you can read about the core, you can read about the investigators and the leads and consultants. But what you're most interested in is to request tissue, access the UW Neuropathology Core Resource Request form. So you click on that. And there's a series of pages. You have to fill out one page before you can click next and go to the next one. So the first page is really mostly about who you are. Who are you? How do we reach you? Who are you working with? Those kind of questions. And then are you mostly interested in the brain bank? Or we also kind of, you come to us for any biospecimens, but some are going to be more maintained by the clinical core, and we work all that out. So this is just where you come if you want a biospecimen, and then we figure out how to get that to you. So then this is just filling out how you want us to send you the tissue. And then this is the meat of it. This is what you want from us. And I'm just going to blow up this part up here, which is really highlighting, you know, do you want the fresh frozen brain? Do you want FFPE slides? And really any number of the fixed tissue, where we go back to the bucket and get fixed tissue for you. And then you're going to give us a description sort of your study or what you're trying to do with the tissue. And again, this might be where we have to follow up with some more specific details so that we can give you the best tissue for your study. You know, we'll ask questions about funding. Just in general, we're set up as a core facility so that we can provide, you know, small studies that maybe don't have funding, or maybe you are doing some collecting some pilot data so you can get funding. We will provide that tissue for it. We don't charge for that to a point. And so it's not necessary that your work is funded, but if we're getting a kind of outrageous request for like hundreds and hundreds of stained slides or something, then we might have to work something out. But in general, we're here to help provide preliminary data for a grant. Or let's say you had an animal model and you generated some interesting findings and you wanted to do some kind of correlational immunostaining in human tissue for a paper that you're going to put out, we would provide that kind of tissue for you. We also will work with people and be on their grants, serve as sort of the neuropathologist, and will be written into people's grants to get a little extra support if what you're wanting kind of goes above and beyond what we're funded to do as a core facility. And then the last page is just what I already said, that that you're going to cite the institution, and then you just submit that and it goes to Amy and we take it from there. So that's sort of the how-to in a nutshell.
Fantastic. Thank you, Caitlin. So let's open it up for questions related to the neuropath core. Let's work through those. And then as we get on toward 1:30, if we want to revisit things and if people have time, then we can kick it back to those other questions. So there's a question from Elizabeth Ray. How often are comorbidities and or drugs taken at time of death listed with the tissue? Are there comorbidities that specifically that would exclude brains from your core?
So I believe the only thing that excludes someone right now is we are really not taking prion cases. We will send those somewhere else. We used to do them, and maybe we will one day. But no, we were one of the few brain banks that was still operating, has been able to continue operating through the pandemic. I mean, there's a real question as to what are the risks if you have an individual who had SARS-CoV-2 to our technicians, to the tissue, you know, what is the exposure? And we've been able to work with people to ensure that everything's done safely for our technicians. So we treat every case like it's, you know, terribly diseased and you don't want to touch it. And so we follow all appropriate safeguards for every case. So nothing will, nothing standard beyond prions will keep us from accepting a case, I believe. And then in general, all of the comorbid sort of peripheral pathology. So someone had hypertension, someone had metastatic breast cancer, all that stuff is like for the ACT study, they have fantastic clinical records. They are the keepers of that. We have some basic information that we know, but if you were doing a study specifically with ACT resources, you would also go through the ACT study and you could get specific details about the clinical, and then we would provide the tissue for that study. So I don't know if that answers the question, but.
Great. I have another question, quick neuropath question. I know there's a delay in accessing samples related to HVAC issues, but probably also COVID. I guess maybe a more general question is, what is the timeline like for getting samples that you request, and what determines whether that's a short timeline versus a longer timeline?
So, right, that's a tricky question right now, and Amy can speak up if she doesn't think I'm being fair, but there's quite a turnaround time because I think most people know our building got shut down and that's where we store all of our frozen tissue. We do have access to that building, it's just under really constraining circumstances. So we have that, and then on top of that, we're in a pandemic with reduced FTE to maintain social distancing. So I think Amy is sending out replies to people's requests that this could take up to 6 months to fill requests. I think we're trying to reduce that so that it actually isn't the case. And under normal circumstances, that is not how long it takes. I think, I mean, weeks to a couple months might be more reasonable. Occasionally someone suddenly needs something and they have a grant due, and or maybe they wrote a paper and for the reviewer's response, they need to have certain, you know, human data that they can incorporate into a response. We will work with you, but there are limitations. We try to keep people in queue and not have, you know, people, you know, not giving preferential treatment, but we will do our very best if something came up and we sort of have those conversations with the investigators. But we do ask for right now, don't hesitate to ask for tissue, but we'll be upfront with you as to how quickly we can fill it. And also it depends on the complexity and the amount of work to fill the request. Sorry, that's like a really wishy-washy answer. But.
Oh, these are wishy-washy times.
And then just, if I could just, thank you, Caitlin. I think that's, I mean, we are trying very hard. For success of everyone, success for the requesters and success for our team. I mean, it really, think way ahead of time because it can be a lot of back and forth. There are a lot of intricacies. related to data, what's related on some, what's related on others. As Caitlin just went through, there's different protocols with how the tissue has been requested over the last several decades, which may or may not meet your needs. So really, I would try to plan 3 months at least ahead of time when you submit your requests. And besides that, we are really working diligently. We are supposed to be back in Harborview, back in the Research and Training Building in February, which will make things a lot easier for everybody. Fingers crossed that happens. But yes, I have a long, oh, and then just one last thing. At any given moment, we're working with several dozen tissue requests. So this is a very, very busy, robust program, very exciting, very state of the art, but very busy too. So
yeah, so if you are thinking about a project and there's even an inkling in your mind you might want human tissue, then yeah, get that request in.
All right, this from Annika Noreen. 2 common questions I get from people wanting to make tissue requests. Are there charges for the tissue samples? And what ballpark sample number for brain tissue is a reasonable request?
Right, so I kind of answered, I think the first part is generally we are here to support kind of, junior investigators trying to get their projects up and running. You need some preliminary data to get your grant out the door, or finishing up a paper and want to have some human tissue as a part of that. So in general, we don't charge for kind of these small projects. And I don't know if Erica or Amy, we did come up with a number, I think. Did you give that number or a ballpark?
For a reasonable request? I don't know. That is sort of like a number that is in flux, I would say. It sort of depends on the number of cases versus the number of slides versus the number of
stains
regions versus the number of stains. So there's no hard and fast number for it because everything is a little bit different.
Right.
I think everyone on here is an academic researcher. We do charge for-profit companies like biotech companies for their tissue requests, but generally do not charge nonprofits.
Right. And yeah, it is kind of a case-by-case thing. And that's where, you'll put in your request and maybe it's a pie in the sky request. And we're not, like I said, we don't dump requests because we think, oh, this person's asking for the moon, we're not going to work with them. We'll call you and we'll say, hey, this is a little bit much. This is kind of more what we can work with. And why don't we start here and then see where this goes? Or we might say, do you have funding? If you can provide funding, we can, do this amount of work. So it is sort of case by case, but that's sort of the general approach.
All right, here's a question. It says, sorry if I missed this, but does the core collect peripheral tissues, they say e.g. GI, liver, etc., for studies that connect changes in peripheral pathologies to progression of AD neuropath? And maybe a broader question is, do you guys do like custom collections? So for example, our group is interested in choroid plexus. Can we get that?
Right, so in general, we do not collect peripheral tissues. Every time I present to a group of pathologists, especially if George Martin is on the call, he makes the plea for more general autopsies because yes, there's so much we're missing by not linking the CNS changes to the periphery. And there are actually a few rapid autopsy programs at the U. The one that comes to mind is the prostate group, so they will collect rapid prostates, but they're not collecting on brains. And so there is an interest group or like a working group that's coming together now to start talking about doing rapid autopsies where they would collect more tissue, but that is not what we do in the neuropath core. However, we have on occasion, we'll get a case suspected for OPMD, which is a neuromuscular disease. So we have to get consent. We cannot just like start sampling people in random places. We have to make sure they're consented for that. But then we might take muscle from a case like that. Or if there was a question about something in the eyes, we have been known to take eyes on occasion. But all these things you have to get consented ahead of time. The one thing sometimes we will do is do a skin punch to get fibroblasts from the skin to compare it to the ones that we're getting from the meninges. And then as far as and then we do get spinal cords, not routinely, but we're doing them more and more with some of the genetics cases that involve the spinal cord. So ALS or the spinocerebellar ataxias, we'll get spinal cord. For specialized requests, we will work with investigators if you need something taken at the rapid autopsy so that you have a specific region that we don't routinely sample for fresh frozen. We can work with you on that. If you need it put in a different kind of fixative, we will work with people on that. Usually it's sort of in a pilot study kind of way, and we coordinate that with you. And it really has to be set up in a way that it's not going to impede the general workflow of the rapid autopsy, which hopefully you got the sense it's pretty involved with what we ask of our tech teams. And so they need to be able to do their job, but if they can do something in addition to help your work that isn't going to impede that, then we try to work that in.
Great. Here's another question. Follow-up to the separation between ACT clinical info and maybe more generally clinical information and tissue samples. Does each brain sample that comes through the longitudinal study continue to be linked to the sample IDs and variables from its parent study, or is there a de-identification step?
So we give them their own ID when they enter the brain bank. They get our code, but we keep a crosswalk so we can always, it's a safeguarded database. So they are de-identified, but we can cross it back and then request the clinical information from the clinical teams, and vice versa. The ACT study, maybe you go through them and you want a certain type of cases, and they'll give you who those people are with the ACT IDs, and we can cross-link it to our code and give you the brain samples from those individuals. So we can always link it back, but it should only be someone who has access to that database that can make that link.
And so I just want to, can you hear me?
Yes.
Okay, so this is also coming up in a future one, will be data management and statistics core. And we have a central database with a central ID. So an individual that has information in more than one database is linked by this unique, totally random ID. That's the center ID. All the data from the ADRC gets compiled, or not all, a lot of the data gets compiled into the central database. Mm-hmm. So yes, it is linked, but it's de-identified. But once you know you have one ID and you say, this is the person ID that I want, then you get all the information related to that person.
Thank you.